Hello, this is Michelle, and I am finishing up the lecture on metabolism. So in class, we talked about glycolysis. And remember that glycolysis is the breaking down of glucose, the oxidation of glucose. And remember that oxidation in this case does not mean with oxygen. We do use oxygen later on when we talk about the Krebs cycle or the citric acid cycle. So oxidation is just the gain or loss. Actually, it is the loss of electrons or hydrogen atoms to other elements. So the end product of glycolysis, you should know, would be pyruvate. And there's actually 2 pyruvate molecules that are produced for every 1 glucose molecule that is broken down. Now, pyruvate could then be if oxygen is not present. So this would be under anaerobic conditions. Okay. This could actually be converted into lactic acid, or we didn't talk about this, but also ethanol. So ethanol can also be a byproduct of fermentation. So this would be fermentation. Now we talked about lactic acid as being something that is produced under conditions, anaerobic exercise conditions in your muscles, and that needs to be broken down even further. And so lactic acid then goes and gets broken down even more. If oxygen is present, then we go into something that is called the citric acid cycle. And the citric acid cycle is aerobic. So that means that it requires oxygen. And it is going to completely break down the organic molecules, and so that the end product is going to be only carbon dioxide, water, and energy. And so this is the kind of the end product of cellular respiration. So you remember that when we talked about this, that this citric acid cycle actually occurs in the matrix of the mitochondria. So mitochondria have a characteristic internal membrane, which is called the cristae, and has lots of folds in it. And the cristae are really important, and the citric acid cycle is taking place within this matrix. And so the oxygen that we breathe in gets dissolved in the cytoplasm of the cell and then gets transported into the mitochondria. So oxygen is used inside of the mitochondria, which is actually quite amazing, because remember how we talked about how the mitochondria were once probably thought to be a separate organism that became incorporated into us. And so this is the chemical reaction, this is the metabolism that is the reason why we need to take in oxygen, and also why we can efficiently produce ATP. This doesn't produce as much ATP, this produces a lot more ATP. Okay, so I mentioned that previously. So if we look at the citric acid cycle, I'm going to fast forward here. This has another name. So the name of this that you'll see in some books or sometimes is called the Krebs cycle, and that's after the person that discovered it. The citric acid cycle is after the fact that the pyruvate is converted to citric acid, and then it undergoes further oxidation and produces high energy molecules that can be then used to produce ATP. So when we look at the pyruvate starts out and it has to be converted into a molecule that's called acetyl-CoA. And pyruvate has 3 carbons, acetyl-CoA only has 2. And so right on the bat, right at the very first thing, we lose carbon dioxide. Now when we break this bond, we're oxidizing pyruvate, and so we can also reduce another high energy molecule, which is called NAD, and NAD can be reduced to NADH, and NADH has a more energy than NAD. Okay, So that is an important aspect of this. Now, acetyl-CoA comes into the citric acid cycle, and it combines with another molecule, which is called oxaloacetic acid. So this is actually plus the carbon that is in the acetyl-CoA. So this is plus the carbon chain, and then the CoA, the acetyl part, CoA, goes off. So acetyl-CoA is kind of released. We'll just ignore that part, sorry. Okay, so this combines. Now, oxaloacetic acid has 4 carbons, and so when we combine these two things, we have a 6-carbon molecule, and this is this is citric acid. So this is citric acid. So when you think about fruit, right, this is citric acid. This is the same acid that is in fruit. Now what we're going to do is we're going to take carbons off of this repeatedly, and then we're going to then produce more high energy molecules. And so if we look at this process, it is going to form a cycle, and it's going to end up with oxaloacetic acid as the very last thing in the cycle. So when we look at this, we can see that NAD positive, which is not very high energy, can be converted to NADH. And this actually occurs 3 times. So I'm going to put times 3. So in the cycle, that happens 3 times. We can also release CO2. So this actually happens 2 times in the cycle. And so you have to think of this as just a series of conversions that are catalyzed by chemical enzymes. They're chemical reactions, and we're not going to go through each one, but I just want you to see what, as we cleave carbon off, we're producing energy and we're releasing carbon dioxide. Okay, so then we also have another molecule that we haven't talked about, which is called FAD, and it actually reduces to FADH2. So it actually becomes more energized with the FADH2. And then we come to the end of our cycle. So one pyruvate goes through this cycle, and then the other pyruvate has to go through the cycle. So to totally oxidize one glucose molecules, you have to go through this cycle twice. So in your book, there's a really nice diagram that shows this. So this shows the 3 carbons of the pyruvic, also shows that we release one and then we get 2 carbons. It doesn't show the formation of citric acid, which is, remember, this 2, right, plus the 4 in the oxaloacetic acid, but it does show the things that are outputted. And one thing that I didn't put in there is that there is one molecule of ADP that is converted to ATP. So let's go back to this. And so we can have this occurring in the cycle. So ADP produces 1 ATP. Okay. Now remember that ATP is a really important molecule that can be used as a universal energy throughout the body. These are not so, and we actually need to actually oxidize them and release energy. It's kind of interesting because as we get older, we actually have less of this molecule, and so it's actually thought to be possibly a source of aging. And it was really interesting because when I was reviewing this information for my lecture on my Facebook page, there was a advertisement for an anti-aging drug. And this anti-aging drug is a precursor to NAD. So the idea here is that as we get older, our mitochondria are less able to produce energy, and so our cells start to deteriorate. And so one of the ideas is that this NAD, right, can be actually used as a supplement, and it's actually been passed by the Food and Drug Administration as a supplement, not as a drug. And so the idea here is that if you take this, perhaps you can boost the energy output of your mitochondria, and your cells will not age as fast, and we could possibly live longer. And so the idea here is that we really need to convert and get back to, right? We need to get this to go the other direction. And that is what is referred to as oxidative phosphorylation. So what we want to do is we want to add on phosphate groups to the ADP molecule to get some more ATP. And this takes place if we have that mitochondria. This is the outer membrane of the mitochondria, and this is the cristae of the mitochondria. Oxidative phosphorylation takes place across this membrane due to the presence of proteins. So there's actually proteins embedded in this membrane. We talked about those protein plasma membranes that actually aid in creating creating more energy. And so we're going to look at what is called, oh, here's a diagram. So this is from your book. And so this shows that inner cristae membrane. And then this shows some of the enzymes that aid in producing energy. And we're going to talk about this. So this is the citric acid cycle. It occurs in the matrix of the mitochondria. Okay. Notice that NADH can actually transfer its electrons to these proteins. And as the electrons are transported, this is called an electron chain, hydrogen ions are then pumped out. Okay. So we can look at what is referred to as the electron transport chain. Okay. So the electron transport chain receives electrons and hydrogen atoms from NADH. So when NADH is broken down, when it is oxidized, so it loses hydrogen atoms, it produces 2 electrons, releases 2 electrons plus a hydrogen ion. And let me just look at my notes, plus a hydrogen ion. There's something else, I think. Nope, that's it. Oh, plus, oh, sorry, plus NAD positive. Okay. So this would be oxidation because it's losing electrons. So remember that oil, oxidation is losing, okay? So what happens is that we have these proteins in our plasma membrane. The proteins pick them up. So the proteins pick up the electrons and move them. Now this is energy. Okay, so some of this energy is used to pump hydrogen ions. So these hydrogen ions are pumped. Well, but maybe not pumped because that implies the active transport, but hydrogen ions are moved to a higher concentration outside the matrix. Okay, so notice here, this is the movement of the electrons. One of the things one of the proteins is called coenzyme Q. Sometimes you hear that when you talk about supplements. One of them is called cytochrome C, right? So there's these different proteins. And the idea here is that it's just, they're transferred, and as they're transferred, the hydrogen gets pumped out. And so what we get is a much higher hydrogen ion concentration out here than say inside the matrix. And that is important because that sets up a potential energy gradient, okay? So the higher hydrogen concentration outside, so I'll write that up here, higher, you can write it down below, higher hydrogen concentration outside the matrix is a source of potential energy, because it's going to want to move, if it can, back into the inside of the cell. So it's kind of like pumping water above a dam. And so the water sits above here, and then gravity, but in this case diffusion, is just going to take the hydrogen ions back in. Okay, so the idea here is that we have another protein which is called ATP synthase. And this is a really interesting enzyme, right? Because it is actually a motor. And it generates ATP molecules by using the movement. It actually moves like a, spins like a motor. It generates ATP molecules. Oops, ATP molecules. I don't know where that went, molecules. So ATP synthase is a motor, and it generates ATP molecules from the energy of hydrogen ions moving back into the matrix. Okay. So here you can see we're pumping, and notice here that FADH2 also gets oxidized and loses its hydrogen and loses some electrons. And then this arrow just shows the movement of the electrons, but the hydrogen gets pumped out. Okay. So I have a kind of a cool video that shows how a protein could act as a motor. It actually turns and produces mechanical energy that can be then used to produce chemical energy. So if we look at my animation here, oops, I have to look for it. Oh, there it is. Okay, so this is an animation just showing ATP synthase as a motor molecule.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
Okay, so that is ATP synthase, and it's all due to the movement of the electrons along the electron chain. So the question is what happens to the electrons eventually? The electrons have to be taken up by something. And so if we look at what happens to the electrons after they are finished with the electron chain, you can see that the electrons are actually taken up by oxygen. So if we have hydrogen inside, right, we combine that with 2 electrons, right, because we would like to add electrons to our protons to make hydrogen, to make a hydrogen atom, right? And then we combine that with 1/2 of the oxygen molecule, which would just be 1 oxygen. And what this produces is water. So the cool thing about this is that the end product of metabolizing or oxidizing glucose is water and then carbon dioxide. So the water comes from the electrons in the electron transport chain, and the carbon dioxide comes from the citric acid cycle. Okay, so remember that this occurs, this is an example of aerobic respiration. It only occurs in the mitochondria, not in the cytoplasm. And it is much, much more efficient than the production of ATP. So at the end of converting with cellular respiration, we have like 38, or maybe it depends upon the cells, but we could have 36 to 38 ATP being produced. And so we talked about how this is much more efficient than producing energy anaerobically.